This video covers principal component analysis, often abbreviated as PCA. It's one of the classic rotation-based techniques. It's an ordination. I'll explain to you what that means. It's based on rotation, so we'll figure out how to do matrix rotations, and then we'll cover a bunch of vocabulary, so eigenvalues and eigenvectors. Those are important to understand in the context of PCA, and then we'll see how that can be applied. So we're looking at objectives, applications, assumptions of principal component analysis, and there's a whole bunch of other vocabulary. That's only going to be in the first couple of videos, but as we start out with multivariate techniques, there is some new vocabulary that is good to get under your belt. So let's just jump right into principal component analysis, and I'd like to locate what we are doing, where this sits in the overall toolbox that we develop as part of this class. So it's here. The primary objective that we cover today is reduction of complexity of big complex data sets, and it's a classic technique, so it's rotation based. So what that means that the scales of the variables matter, and you can use it for other purposes here, so we'll get to this another day. But we'll simply start with working with one data set and see what we can do in terms of complexity reduction. So when we talk about multivariate data set, those look usually like this. So it's a standard data table. I'd like to have an ID column here, and each row represents typically a sampling or experimental unit. So those can be individuals, or they can be plots or transects, or whatever kind of data you collected, maybe a core from a borehole or from a tree ring sample, something like that. So each of the rows represents an independent sampling unit. Those sampling units can often be grouped, so you can group them by locations or treatments or ecosystems. So those different colors here, this may be forested ecosystems, this may be a grassland ecosystem, or maybe these are different species. So the blue species, the green species, the red species, and so on. So oftentimes you have some sort of structure. It can also be an experimental structure. So this could be treatment A, treatment B, treatment C, and you want to then see what happens to multiple variables. And your variables can be also anything. So this can be species frequencies if those IDs are plots, maybe plots in different ecosystems represented by colors, and then you can look at a species 1 and see what frequency those have in those different plot locations. Or it may be soil parameters. Well, maybe these are soil samples that you collected at different locations. It could be climate variables, morphological traits. So if these represent species, maybe you want to figure out what the different species characteristics are. So is the red one more drought resistant than the green one, and so on and so forth. And again, these can be many variables that describe a species. So those could be morphological traits, physiological traits, genetic markers. You can apply this to virtually anything. This could be spectral bands from a satellite. So these individual observations could be individual pixels in the landscape, and then you have certain values for different colors in a multispectral satellite image. And so those can be very large data tables, so you can easily have dozens of spectral bands or even more. You could have dozens of species in a community ecology study. You can get hundreds of genes or dozens of traits. And so if you have these data tables, it really is hard to make sense of those. So in order to wrap your head around what's really in your data set, you need to reduce complexity here. So if those data tables are hundreds of observations long, or thousands or even millions in case of satellite data, and you have all these variables, how are you going to wrap your head around this? So this is where multivariate statistics come in. So one way to reduce complexity is to see if variables correlated, and that's exactly what the principal component analysis does. And I can visualize this simply by plotting one variable over another, and I see these variables are nicely correlated here. So if they are nicely correlated, then I can collapse those points onto the axis here. I'm just going to move them right onto those axes. So now if I take this axis and put it here, then my points would sit right on this axis. And that new axis here, I'm calling this variable 1 and 2. So it's a combination, and I could also call this my principal component 1. So this axis here, pc1, so it's a kind of combination axis, becomes a new linear combination of my original variables, representing variables 1 and 2, which were nicely correlated. And that actually gives me some freedom to plot another variable here. So let's put variable 3 here. So I now have some space in my two-dimensional plot, and I can now locate where those points would be. Maybe now I have something like this. Maybe those ones sit up here. So something else, another dimension in my data set has just become apparent in a simple two-dimensional plot that I couldn't see before. So this one represents variable 1 and 2, and this one represents variable 3. So instead of having just 2 variables in a scatter plot, I can have 3. So that's complexity reduction. That makes things easier to look at. So I don't need to look at variable 1 and 2 separately, because they are highly correlated. They basically mean the same thing. If I know the value for variable 1, I also know the value for variable 2. I don't need multiple plots, so let's make it simple. And if I have many variables that are well correlated, what I hope in the end to get is a situation like this, where my principal component 1 represents a number of variables, and my principal component 2 also represents a whole number of variables. So that's all good, but how do I actually do this? How can I collapse these variables into a few components that represent multiple variables? You know, if I have perfect correlations, that's easy. So I could collapse all the ones that are perfectly correlated into one component and all the others that are perfectly correlated into another one. That would be the simplest thing, but that's not how it works. So it's a much more messy situation. Each variable is usually somewhat correlated to another, some more than others. So it's a slightly difficult problem to actually implement, although the idea is good. And the way that this is done is with rotation, with a rotation of a coordinate system. So that's a classical approach to multivariate statistics. All I do is rotate a coordinate system with multiple variables. And here we have a classic example that's widely used in R. So I plotted this here as a 3D plot. So we have petal width, sepal length, and sepal width. And we can rotate this coordinate system so that if we have variables that are highly correlated, that they get collapsed into one axis. So we can do this rotation by hand in, you know, either Python or R. We can just do a 3D plot since we only have 3 variables, and look at it from different angles. So this is really what you do in principal component analysis. And let's do a rotation so we can look at it from different angles and see if that reveals anything that is interesting. Often what you do in principal component analysis is that you try to line things up in such a way that you have a horizontal axis here that explains most of the variance. So once we have that, so once we have this data set stretched out as much as we can from left to right, then we are going to rotate it in the second dimension, so around that first axis. So rotate it around the first axis so that you get maximum separation of your data points from the top to bottom. So that might be a good principal component analysis that we have done here. So in this particular case, our x variable here would summarize primarily sepal length, so that's well represented. And my second component here, that represents sepal width, But also, this axis here also lines up with the vertical axis quite nice. So petal width and sepal width are probably somewhat correlated. So that second principal component now summarizes two of those variables. Now, obviously, I didn't do a good job, but you can do math to get this exactly right. You can do other math that has another objective. So I may not be interested in explaining the maximum variance here, like we do in principal component analysis, but I may want a rotation that separates my groups best. So in this case, the blue group is nicely separated, but the red and the green one here, they are kind of crowded on top of each other. So that's not a good way to look at my data if I'm interested in differences between species or between ecosystems based on multiple variables. So let's rotate this a little more, and maybe that is actually not a bad view. So now let's see, yeah, maybe like that. Now I have my three groups nicely separated. That's actually called now a discriminant analysis. So it's the same principle, but with a rotation with a different objective. And I can see petal width is a variable that is very useful in distinguishing between those three species here. So I can have different objectives in looking at my data set from different angles. And so those define actually what your particular techniques are. So if I rotate things so that the maximum variance is explained in my entire data set, that's called a principal component analysis. And so in that case, you don't have groups, or if you have groups, you are just ignoring them. They're just coloring your points, but they are not part of the math that you're doing. So just to recap what I've shown you before, you try to find a principal component that maximizes the variance explained in the data set. So you want to stretch it out as much as possible in this horizontal direction. And then once you determined your first principal component, you fix that, but you can rotate your cube or your multivariate coordinate system around that first axis to get a second principal component. So if we do this, so here we can't really see much spread up and down, but maybe if we rotate this cube a little bit, then we see another dimension. So instead of a line here, this may have actually a curve. So I'm just looking at it from a different angle, rotated around this horizontal axis. So your second vertical axis is your principal component 2, and so on. So once I have those two fixed, I can still rotate it in more dimensional space, so I can't visualize this, but you have as many principal components available as you have variables. But the objective really is to look at your data set in such a way that most of the variance is captured in as few dimensions as possible, preferably 2 or 4, so that you can represent it on a flat piece of paper with 1 or 2 graphs. So that's principal component analysis, and also factor analysis. Factor analysis gives this a slight twist. So I like to think about it as a PCA, but with that maximum variance explained, not taken too seriously. So it does some additional small rotations that make it more interpretable. But there are other ways you can look at things. So if you want to maximize the variance between groups, just as I've shown you before, then that's called a discriminant analysis, or, interestingly, a multivariate analysis of variance. So that's actually the same thing. You're just looking at different aspects of this. But mathematically, these are identical. And if you have two data sets, you can kind of rotate two cubes. So this would assume that, so if we look back here at our data set, that I have a bunch of variables that belong to one category, let's say species frequencies, but then you also have another set of variables that you collected here, maybe climate variables or soil conditions. Oftentimes your multivariate data set is composed of groups of multiple variables. So if you have a situation like this, you can rotate those groups separately, so I can line up my species frequencies and my soil variable, so I can rotate those two groups independently. So if I have two sets of variables, I might want to rotate them in such a way that the correlation between those are maximized. So then I can take this component here that represents my species frequencies and this component that represents my soil variables and plot them one over the other. And that allows me to say, okay, soil variable 1, 4, and 7 influence the species frequencies of species 3, 4, and 9. So again, I've simplified this situation, but it's also an inference. So those are called canonical correlations or redundancy analysis, canonical correspondence analysis. They work all based on those principles. So what I've done here is also called an ordination. So if I have a large number of variables and I represent them in 2 dimensions, so my variables are my axis here and my sampling units or individuals or experimental units, they are represented by those dots. They may belong to different species or different classes or different treatments, but they don't necessarily have to. That's called an ordination. And I'm just going to show you how that really works. So I simply rotate my data set in a position that I like, and then this really is my ordination here. So this point of origin, that is carried over, so I'm setting this to 0. This whole data set is normalized, and well, let me draw some points here. So those are my red ones, those are my green points. I'm just putting a few here on top of what we already have, just to illustrate to you how this works. And then I'm going to look at my individual axis here. So that one was sepal width. So I'm calling this sepal width, and this one here, that is my petal width, and that one here is my sepal width. Very nice. And now I can delete this, and I have a proper ordination. So this is my PC2, this is my PC1. So they both explain a certain amount of total variance that's in my data set. So let's say this one explains 54%, and this one explains 23%. And these numbers here, they're actually related to the eigen values. These vectors are these original, this shadow of my original coordinate system. Those are the eigenvectors. And we've done this here with a 3-dimensional system, but there's no reason why you can't have other vectors here. For example, if we need the petal length that we couldn't include here, a 4-dimensional system can also be projected on 2 dimensions, so that is not a problem. So that's how all ordinations work in principle. So it's a simplification of a complex data set with many variables and many points, and you can make sense of this quite easily. So all points that are at the bottom have low values for petal width and petal length. So you can see the blue group that would have low values in that case. The green group would have high values, and the points that are to the right, they have high sepal width and low sepal length. So that's how this works and how the ordination is interpreted.